So good afternoon to everyone. We're going to be speaking in English. We're actually recording these presentations, so I'm glad to see. Has all of you people come last time? How many of you were here last time? Okay, so a good number. That's very good. So now do I have newcomers this time? Any newcomers? Okay, a few newcomers. Okay, for you newcomers, you can request the past the recording and get up to date, all right? So I hope it will be of benefit. So let's start. This is a part 2 of a 3-part series. We're going to be talking a little bit about the elements.
The broadcast is now starting. All attendees are in listen-only mode.
Okay, so this is the part 2. And let me figure out. Mariela, que yo le doy aquí para que se mueva esto. Okay, so yes, just to get an overview of the part 1. Last time we spoke about the understanding of the publishing process. We talked about the biomedical scientific publishing, what and when to publish, the literature, the importance of commitment, deciding the important subject of deciding on a target journal, and the new issues of giving the public access to scientific publications, and the very controversial issues on co-authorship. We also started to talk to you about preparation of a manuscript. We gave you the conceptual framework, and we began explaining about the title, the keywords, and the abstract. So now we're going to go diving into the basic 4 elements of the manuscript, right? So if you look at it and compare to an experimental process as to the section of the paper, so when you answer a question, what did I do in a nutshell, that refers to your abstract. What is the problem? That's your introduction. How did I solve the problem? You answer it with the materials and methods. What did I find out? That's your results. What does it mean? That's what you write in your discussion. Who helped me out? That's what you give credit to in acknowledgement. And whose work did I refer to? Is all the citations you have in the work. Extra information, some journals accept it, some don't. You put in the appendices. So we're going to start now with the very important subject of the introduction, and I'll leave you with Dr. Gonzalez to cover that subject.
Good afternoon to everybody. So I guess that everybody has a favorite paper. If you think about the papers you've read, you have the super boring ones, the ones that actually capture your attention and you could actually name one of the authors or at least part of the title, and others that you simply just like skim and decide not to read at all. Today we're going to give you the tools to become one of those papers where people actually remember your name when they read them. So good data tells a story. A good paper will lead you one by one by one till you actually get the whole aim and focus of the paper. It'll hook you and it'll lead you in, and that's what we want to do. These are all the parts of a paper. All of these are your tools to tell your story. The title has to convey the focus of the study and attract the reader. If you fail there, nobody's going to read your paper. The abstract just gives them a little taste and gives them the main points so they could go into your introduction. The introduction is one of the most important parts of your paper. If it's not written well, the reader will not understand your work or your data. The materials and methods provides the details. The results describes the findings. You need to think about figures, tables, graphs, which you will use. The discussion discusses what the results mean, and then you have your references. Just like Dina mentioned before, I just wanted to say again, these are your tools. When you're thinking about writing a paper, you have to think of all the tools necessary. These will vary according to the journal. You always have to go to the instructions to authors page, so they give you the exact sections they want you to include in the paper, but these will be your tools to tell your story. So what is the role of the introduction section? So the introduction should give the readers the information they need to understand your paper without having to go read other publications. How many of you have had to read an introduction and go searching for the references? I have, because I really don't understand what they're saying. That's exactly what you don't want to do. You don't want to give too much information so they lose track of the focus of your paper, but you don't want to give them such little information that they have to go looking for references to understand what you're saying. So the introduction should provide the conceptual justification and framework of the research. Why is your research important? What is the gap in knowledge? Why is this information important within your area? Emphasize how your research may advance the field. So in terms of organizing the introduction, there's no concrete blueprint you can follow, just recommendations. The first things I do is I look at what the contextual orientation is and what's the manuscript focus. These are the main two parts of your paper. When you think the introduction, when you think about introduction, it should approximately take 2 pages, double spaced, at most 8 paragraphs, ideally 5, 4. So when we think about the contextual orientation, which is a brief summary of the current status in the field, think about the first paragraph. You should go from general to specific. So the first paragraph, you should take advantage of epidemiology, any sort of statistic that brings importance to your field. Say you're studying a disease, you're studying a bacteria, talk about how many people, the incidence, the death rates, talk about how many people are infected by your infectious disease. Just give a general context as why is this important. When you go down to the second and third paragraph, maybe, you want to narrow down to exactly what you're going to be studying. You want to describe what's known in the field. You want to state what's missing, what's not known, and then state why this information is important. Then after that, you shift into the manuscript focus, where you give a brief overview of what you did to address this gap in knowledge. What you're doing should address the gap in knowledge you mentioned. If not, the gap in knowledge you're mentioning before is incorrect. Always check that that is concordant. You should state your hypothesis. What were you expecting to observe in your study? And end with a powerful sentence. Why is this important? It's very, very important that you never, never, never sell the outcome of the story here. You don't want to give the reader the reward before they read the whole paper. You just want to draw them in. So before you start, when you're trying to write a paper, you need a muse, you need inspiration. I know that everybody that has written paper, you start and then you come with a writer's block. These are some of the things, at least I do, to try to get over my writer's block. One of the first things you should consider is where you're submitting. Are you submitting to a specialized journal or a general journal? Why is this important? Well, if you're submitting to a specialized journal, say Helicobacter pylori, you don't want to really go into all the details about what type of bacteria it is, and is it a gram-negative, gram-positive, because it's based on that bacteria. Readers that read that journal already know this information. Whereas if you submit to a journals such as PLOS ONE, maybe you do want to go into this, because you'll get a variety of readers that do not necessarily know all of this background information. One of the things I find most helpful is that I look for a manuscript that I like. It doesn't have to even be in my field. I just want to see what the author, what his thought process was. If I liked it, it probably tells a story, and it probably does it well. So I look through how he goes through the thought process, and I try to do something similar in mine. You could also look for manuscripts in your field that describe similar results. They could also be very helpful just to give you an idea of where you could start. And then, before you start, you should really be clear in terms of what's the single most important piece of knowledge or conclusion that you provide by your manuscript. This will dictate the spin you're going to give on your manuscript. And one thing to remember, it's in the bottom of the slide, is that when you write a manuscript, it's always in past tense. So have that in mind. So let's try to go a little bit more into detail in terms of each paragraph in the introduction. In the first paragraph, you should always, just like when we discussed last semester, when writing proposals, you should start with a sentence that grabs the attention of the reader. Because if you start with a boring sentence, odds are the person will not read through your paper. You want to have a sentence that's impactful. You should really not use very general information. I just gave there an example. Breast cancer is a deadly disease versus breast cancer is the leading cause of death among Puerto Rican Hispanic women. I would much rather read the second than the first. And these are things you need to think about when you're writing down the elements of your introduction. You could also, as I mentioned before, provide an overview of the disease, condition, organism, define the big picture, use epidemiology information, anything that supports the significance of the field you're studying. As I mentioned before, specialized versus general journals. This will dictate how you shape your first paragraph. So in general, H. pylori, I just gave this example, is a gram-negative bacterium found in the stomach versus the second sentence, which is a little bit more specialized. And that's just a general example, but you should think of all of these things before you write your introduction. In the second or third paragraph, you want to narrow it down to your field of interest. These paragraphs should go from a status report of the current knowledge in the field to the gap in knowledge that you will be addressing. One of the things you don't want to do is to comprehensively review the literature. This is not a review, unless it is a review, and that's a whole other story. We're talking more about presenting your data, an original manuscript. You don't want to do a comprehensive review of the literature. You just want to include the literature that's relevant to your work. And you can't cite your earlier work. Actually, it is recommended if it supports what you're going to be presenting. When you address the gap in knowledge, you have to convey it in a way that highlights why addressing this research is important. And how important is stating this gap in knowledge? Well, it's the second most important factor in terms of article acceptance or rejections. So this is an example of how you could phrase the gap in knowledge. You can feel free to read, but I'll read it out for you. While there is a considerable evidence to support the conclusion that genetic ancestry may increase breast cancer risk, the actual underlying mechanisms are yet to be elucidated. The are yet to be elucidated is just a way of sort of hinting at the gap in knowledge. You could also use are currently aren't clear, they remain to be identified. You could use the phrase of your choosing, but it must be clear that this is missing in current literature. A comprehensive understanding of the mechanisms by which genetic ancestry contributes to increased breast cancer risk is needed in order to develop novel strategies to identify women at higher risk of breast cancer. This is the importance of knowing this gap in knowledge. So this is just to give you an idea of how you could phrase the gap in knowledge and the ultimate advancement to the field. So the fourth and fifth paragraph, now you're going into the nitty-gritty of what you did. You want to clearly describe the aim of your study, and you should use the aim of the study was. You don't want the reader to guess what the aim of the study was, because they can get it wrong. And you should describe the overall approach used to address the gap in knowledge. You don't want to go into details. X micrograms of DNA, blah, blah, blah. But you want to mention, if you use an in vitro system, what model system did you use? Was it a case control study? All these things in a couple of sentences could describe in general what was done. You want to state your hypothesis in a way that's conveying to the reader clearly what you were expecting. And this is optional, but I really like to include this in introductions. State the impact on the field of what you were studying. How does it advance the field? And you could state the overall finding. Again, you don't want to go into details regarding the results and the conclusions, but you can just give them a hint of what they're about to read. This is an example. This is a longer example. But if you go to the last two sentences, it's clear that this is an independent case control study. It gives you the number of participants. Sometimes this is really important to say because maybe the gap in knowledge was that similar studies had lower number of participants. Therefore, this study will have a higher power. And it tells you that they did this type of study to investigate possible interactions between 3 single nucleotide polymorphisms in H. pylori infection. So when I was looking for this, I tried to find a last paragraph that encompassed all the things I said in the previous slide, and it was particularly hard. So that gives you an idea of the flexibility there is on how to write it and the things you can incorporate to make it even better. And now I'll leave you with Dr. Holman.
Okay. Any questions so far in introduction? No? All right. So let's go to materials and methods. This, what you really need to have here is introduction is more an elaborate, creative thinking, creative drafting, and creative writing. Materials and methods is you have to be meticulous. You just give the significant, sufficient technical detail to be able to reproduce the study, okay? It's very important that you allow others to do the same thing you did and come out with the same results. So you describe everything used and done. A lot of people believe it's the boring part, and it kind of is, but it's the easiest part for most. It's straightforward. You don't need to be a super intelligent person to do it, but you have to do it right, okay? So if you compare it, it's just like writing a recipe in a cookbook, okay? You need your list of ingredients, that's your materials, your step-by-step instruction, those are your methods, and you need to, you can generate the dish, which is the results. If you leave out a special ingredient, we don't need no mystery components like in McDonald's, whatever, the Big Mac, the mystery ingredient, or in Coca-Cola, the mystery ingredient. If you leave out something, no one can reproduce your results, and it's not considered scientifically ethical, okay? So you have to list everything. Now, when a good rule of thumb would be is delay it after you get your results. Sometimes you give you write it up front, then along the way you change what you did, and then you wind up with the materials and methods that really doesn't look like the results. So if you want to sort of avoid that, get your results, you have your results already, and then go back and look through it, and then list everything you have there, all the reagents, the methods, the equipment. If you're talking about group studies, then what are the characteristics? Organize them into different subgroups, make sure you have everything there. What are your cell lines, antibodies, tissue culture reagents? There are many out there. You want to make sure you wrote the same one, the correct ones, and summarize them. And always past tense, never in present tense, because you already did this, okay? So you're reporting what you did. Then you describe everything in detail. You need to list your buffers, solutions, sources. There are many different. You want to make sure that anyone can understand and reproduce what you're doing. If you have a unique material, reagent, or data set, you have to really emphasize what is there. The source, if someone donated something to you, you have to mention it and what institution they come from, okay? It's very important. If you're using the pharmaceutical source, if you're using a new drug, investigational new drug, that's very important. Actually, it's very important. So if you have unique materials, if you're doing RNA, etc., cell lines, cultures, and for animal strains, they even require that you explain the age, the sex, and what maintenance issues these little animals had beforehand. So a person can just go through that and know exactly how it was done. Okay. So then, the methodology, what you want to do is how everything was carried out. Why the studies were done in a certain way, you need to explain, because if you vary from a method, you need to explain why. Why more than one approach? Maybe you tackled this one way, and then you had to repeat it and do it another way. That needs to be explained. And then if it's not very common, you mean to give more detail on that. You could probably refer to earlier publications and give a general view of what the method was, but if you change something, you really have to give more detail. And you decide to change for what reason. Okay, it's very important. I know that at one time when a lab here in Puerto Rico, we tried to copy some methodologies done in the United States, but the environment was very detrimental to how the chromosomes came out. So we had to vary a lot because the humidity here is so high, and the technique and methodology had to change to be able to get those chromosomes all nice and neat. As the frizzy hair you get here in Puerto Rico, it happens with the chromosomes too. I just learned that. That was a while ago, but anyway. Okay, so when you cite the methodologies, everything novel, you have to cite the first time it was introduced, okay, first time it was described. Sometimes you assume if it's common, you don't have to cite it, but remember where you're writing at, because it may be common to you, but maybe not common to your readership. So you have to decide who are you going to write to and if they really know it as well as you. Don't cite like multiple techniques, you're not all over there, just one, the original one. And if it was modified, you really, really have to cite, describe, and explain the modifications. Okay, if you're getting commercially approved, provided assays, just, don't refer it to as the kit, as in this. The assay was done according to the manufacturer's instruction provided with the kit. The readers don't have the kit, so you don't do that, right? Okay, then you have to, it's better to say the ELISA assays used to assess serum levels of X was carried out as described in the manufacturer's instruction provided with these assay kits on triplicate samples using purified recombinant X standards. The assay has a sensitivity of 1.1 nanograms per milliliter. Isn't that more expressive, more descriptive? Okay. So then I know the last conference we talked a lot about plagiarism and how we have some plagiarism police people always checking out these manuscripts and make sure you're not doing that. And it's become a big deal. You can lose your job, you lose the affiliation if you were caught plagiarizing. So and methods and materials and methods, there's only so many ways you can write the same methodology, okay? So if it's similar, and the Office of Research Integrity will not pursue it, because you can't really, you take the Petri, you do this, and you can't really vary so much or become too creative in that respect. So they don't really pursue this if it's very similar. It's not serious, but just remember to cite the original paper, okay? So you won't get into any problem. Then for randomized clinical trials, I don't know how many clinicians are here. There are a few, but you really have to follow the consort statement. And within the materials and methods section, they really want you to address how was the enrollment criteria? What were your inclusion-exclusion criteria? What were in the interventions? How were they done? What was the timing? How long did they take? What the laboratory tests and who conducted them? Is it someone known to do good work? The outcome measurements, the sample size, how do you obtain that number? It's not a magical number you got caught out of your sleeve, right? You reached that sample size based on some calculations. How did you randomize them, and all the stats. Statisticians love this because they write their little paragraphs and put everything there. Okay, but then a few issues came up on controversies regarding your materials and methods. Is that some people have developed very unique materials or software or data or reagents to be able to do the work that they're doing. And then if you look at it, very to the word, it means that to be able to have other people repeat what you're doing, you're going to have to give them that. authors and investigators are going to say, what, I developed all my work with that. I don't want to give it away. So there's a lot of arguments that they don't want to share these unique materials because it might have a potential commercial value, and they're working on their negotiating rights on the patent. They want to protect, this is going to work that's ongoing for fellow students or people getting their PhDs. Maybe it's too costly and too time-consuming. If it's data sets, you don't want to compromise their confidentiality of the patients. And there might be some restrictions on being able to transport these biological materials. So these are all the authors, gave out these, why we don't want to share. But then, National Research Council really did publicate that if you're going to, there must be a universal adherence to full disclosure and unrestricted access to the data materials. Okay, so they're saying that in order for you to publish, you have to assume certain responsibilities, and you have to ensure your peers, which might be your enemies, to reproduce the findings, okay? And you have to disclose everything, including the materials. Even some journals, as I cite below, Nature and Science, did write specific policies that needs to be done in their journals. So if you're uncomfortable with that, don't publish there, okay? Because you're going to be requested to disclose everything and share. Okay, so after these words, let me get you back to the results section now.
So a very important part of your manuscript is obviously the results section. The results section allows the reader to fully assess the experimental evidence on which the authors base your conclusions. It should be written in a clear, concise way. It should tell a story. And that's when it gets a little bit difficult at times. How do you mesh all these different experiments so that the reader can see how you progressed from each experiment in a logical way? And this is critical for the manuscript to be favorably reviewed and to be cited in the future. So where to begin on writing your results? Tell a story. Think about the story. How did you come up with these experiments? How did you go from this experiment to the next experiment? And this will help you how to decide how you will organize the subsections. Determine how you will format your data. Will you use graphs, tables, figures? Do you save space by using a figure of a table? You don't want to include a figure if you could tell the results in one sentence, for example. Is the concept clearer to the reader if a figure or table is used? And then there's the do's and don'ts. You do want to present essential experimental evidence, but many times we've done so many other experiments, but if they don't support your conclusion, you should not include them in this paper. Share a brief description of the experiment and describe the rationale. This is a definite do, but do not repeat the method in detail. You want to just give them the overview of how it was done. Present your results with statistics. Don't present your raw data. Mention the figure table in the order of they appear and place them in a clear logical order. Provide standalone figure graph table legends. Table legends are a must. Try to avoid the vague figure graph table legends. Provide a brief interpretation of the data. Do not add conclusions or discuss the results. Include supplementary data if it supports the conclusions. This will depend on where you're publishing. There's lots of journals that now do not accept supplementary data. You should always consult with your instruction to authors, and they'll give you exactly what they want and the things you can include. And remember, it should always be written past tense. So when you're thinking about writing your results section, think about including these 5 steps in each of your results. What is the concept? We have postulated that breast cancer risk is increased among Puerto Rican Hispanic women due to admixture, for example. You just start with a sentence explaining why you're going to go down that road and do that experiment. Then you go to the question, this would suggest that. And then you mention the approach. If the approach was, for example, qPCR analysis showed, then you go straight to the nitty-gritty and talk about your results and mention the figure, table, graph where they're included. And then at the end of that paragraph, you include a sentence with your interpretation. These expression patterns suggest X. It's important that you do not draw conclusions in the results and do not discuss compared to other papers. The results in general do not have references. I've seen several papers that actually go into this discussion and add references there. That's generally not how it's done. You just do an interpretation, and when you discuss the results, it'll be in your discussion section. So I wanted to give you an example of what an interpretation is, so there's no room to get confused about this. If you go to this paragraph, was taken from an actual paper. And in the first few sentences, it's describing the results. It tells you that the expression of what gene was significantly higher in X condition compared to the other. But if you go down to the last sentence, this is how a way that you could write the interpretation. These results suggest that the photoreceptor regulates light-dependent expression of this gene. And that is what an interpretation would be. Anything beyond that would be actually discussing the results. So how do you keep logic and flow? When you're reading a good paper, it just flows to the next idea and to the next idea. And this begins from the introduction, goes all the way through the methods, goes through the results, and it's also seen in the discussion. So in terms of the results, these are some techniques you can use to keep it flowing from one idea to the other, leading the reader from this experiment to the rationale of the next experiment. So it's called the hook techniques. You could use the leading hook, the trailing hook, or the repetition hook. When you think about the leading hook, it means that you start a paragraph with the main point made in the previous paragraph. And one way you could do this is mentioning the results shown in the figure in the above paragraph. These results shown in figure X support blah, blah, blah. Therefore, in the next step of the study, or in this phase of the study, we performed X and X to address. So you sort of lead them. So this was my conclusion from the first one. So the next logical step to prove X would be this. The trailing hook, it's totally the inverse. You finish the previous paragraph with a point to be made in the following paragraph. The results shown suggested X, and then you start the next paragraph to prove if X was blah, blah, blah. So it gives you a nice flow from paragraph to paragraph. The repetition hook is not really as recommended to be used, and that's where you use in every paragraph. You repeat the main point. And this could get a little bit sort of tedious. So it could be used interspersed throughout the results section, but the leading hook and the trailing hook would be better to keep the flow going in your results section. So Dr. Hallman briefly talked about clinical trial methods, and results also have guidelines as well. I'm providing the link so you could go into the website. They actually have a checklist that goes from every section of the paper, what you should include in every section. And the concert groups starts for consolidated standards of reporting trials. And it encompasses various initiatives established by this group to alleviate problems that arose from inadequate reporting of randomized clinical trials. If you need any additional information, you could go to the website, and as I said, there's a nice checklist you can follow. So some of the hardest parts of Writing your results is how you're going to convey your data. Do you use tables, figures, graphs? Sometimes you don't even know where to start or how to actually, what would be the best way to show your data. So one of the things that you could do is that you could look at other publications to see how others have presented similar data. I think that one of the things that I find most helpful sometimes is when I talk to the statistician. They have very nice ideas on how you can show different type of data, how you can organize tables, and that's actually very helpful lots of times to get you started on how you can organize your data. Have in mind that when you use visual aids, they should increase the readability and facilitate understanding. If you have a massive table that is just like there, think about it. That doesn't really increase the readability or facilitate the understanding. But maybe there's other ways, like you can highlight or bold the sections you want to actually mention in the text, or bring the reader in to exactly what you want to show them, rather than make them guess at the whole table with all of this data. Also, when you have a visual aid, the reader should just look at that table or that figure and take the main message home. If you do not achieve that effect, you should really rethink on how you're actually showing your data. And one of the most important things that could actually help you take that message home is using an informative standalone legend. You could use a title that highlights the main finding, and that it immediately gives the reader exactly what you want to show, what you concluded from them. You could briefly mention in the text the method used and what you found, and always mention the statistical representations if needed. What was the significance? Was it P lower than 0.05? Did you use some sort of abbreviation, some sort of test? You should always mention it in there if you use them. So I wanted to give you an example of an informative figure legend. I did not read this paper, but if you go to the figure legend, you'll sort of understand what they did. If you look at the title is sort of their interpretation of the data. You could use that, or you could mention it in the legend and use the second sentence as the title. It'll depend on your style and on what you want to do. So in this one, you see that the interpretation is a title. The second sentence is telling you how they actually did the experiment. They tell you A and B, they tell you what they use in each, and they give you all of the abbreviations in the figure legend. You don't have to go to the text of the paper to actually sort of get the gist of what they did, and that's exactly what you want to achieve with a figure legend. How you write it, how you organize it, it'll be up to you, but it should give you the whole information. In terms of supplementary data, so this got really, it started to be used a lot. And it's actually pretty helpful, especially in the really long cases of these tables where they're just ginormous. And you can't really include it in the paper because it's too big and it occupies too much space. So policies vary according to journal. Some journals are actually opting to not accept them. They think that it just gives the reviewers more work and they have to review more. So if you can't include it in the manuscript, they're not really endorsing that you use this. But at the same time, if you want to use movie clips, sound files, this is a really unique way to provide a taste of what you did that you can't really read in the paper, but you can actually see. In terms of a very good definition, Nature defines supplementary information as peer-reviewed material directly relevant to the conclusion of the paper that cannot be included in the printed version for reasons of space or medium. Directly relevant to the conclusion of the paper. As I mentioned before, sometimes you present in your paper 5 experiments or 5 essays that repeated in triplicate, but in honesty you did 10. you don't want to really, if it doesn't support a conclusion of your paper, you don't want to really publish them or have them in your supplementary data. But if some of that data actually drives the point home and makes your data more, your results and your conclusions more solid, well, you should definitely think about putting it in the supplementary data. So I cannot go into results without really briefly speaking about ethics and things you should consider. What about representative figures? Representative figures are sort of sketchy in terms of you can run a gel 4 times and it'll never look the same, even though you use the same amount of DNA and everything is exactly the same. So in terms of using a representative figure, you should be really, really cautious. It should only be included if you repeated it several times and if there's statistically significant results based on the several repetitions. In terms of digital manipulation of figures, the only manipulations that are considered acceptable are cropping, brightness, and contrast, but only if they're applied to the whole figure. You cannot just apply it to an end of the figure. Any other manipulation of a digital figure is not ethical. Some journals have advanced forensic techniques to detect if you've doctored your images. And I personally have had the experience where they ask for the original. So save all of your original radiographs and pictures and have them in file. And try to not manipulate figures at all. The instructions for authors have very strict guidelines on what they allow as digital images. It'll go from the pixels. Most of them ask for 300 PPIs, I think that's what they call. And these should always be followed. So have that in mind. And I think that in terms of ethics, and it sort of gives you something to think about in record keeping. For example, if you're called because you're publishing, like it happened to me, they want a radiograph that you did 5 years ago, you should have that radiograph somewhere where you know where it is, or that original picture so you can provide it. And with that, I'll leave you with Dina.
Okay, now we're going on to the discussion. Now this is the part where, you know, everybody says you sit down, you wrote everything down, and then what am I supposed to write about, okay? So this is like the major challenge, okay, that everybody sort of gets stuck, the writer's block, as Maria said. Okay, so, and then how many topics should I write? Always be concise. The goal is to have at least 2 to 4 pages of a double-spaced Arial font. You know, it's the larger font, more clear, and that really translates to about half or 1 printed page when it's in double column. So that's about approximate okay. But then a good tip would be to write it immediately after you got the comfortable, almost final draft of the results. I know I said before to write your materials and methods after the results. Now I'm telling you to write this after the results, but this is immediately after, okay? Because you got the results right there in your head, really, really fresh there. And then it's easier to comment, and then instead of saying, okay, I need my muse to sit down and look at this, and what am I going to write about? Okay, so what should a discussion do? It should like really build the intellectual bridge and validate the importance of what you've done. It should be synthetic. You don't want to go on and on and on. You know, everyone hates the professor that takes too long to drive a point. So this is what you're doing right here. And you want to give something to all your readership as much as possible. So you have to inspire and expand the field. Again, go back to who you're writing to and who the readers are for that paper. But then also in your discussion, you want to give the leverage that you want for your manuscript. This is where you're saying, this is where you're showing off and saying, look how a good writer, how much of a good writer I am, okay? What you should not do is just recapitulate, write again about the results. That's boring. Okay, repeat the introduction. Of course, you don't want to because you're writing about the importance of the results here. And you don't want to go on and write a review article because what is important is your findings here in this primary peer-published manuscript. Okay? So discussion is exactly where the importance of the results are emphasized. And usually, if you're like me, you can't wait. You look at the abstract, oh, this looks good. You jump right to the discussion and see what they had to say, right? And then you go back and look at that if you want to, the materials and methods and so forth and the results. But most people really like say, oh, this sounds interesting, this abstract. You go to discussion. If it's really good, then you look at the paper. So take that in mind. The goal in discussion is to give the answer to the question that you rose in introduction. You want to briefly review the findings to generate this answer, and you want to convince the readers that it was important. This answer was important. Okay, so the first paragraph, as Maru explained to you, the first paragraph is your hook. So you want to bait that hook by saying, and from the very first sentence, is the most important finding should be, really write it out. It should be the answer to the questions. Then after this first sentence, you can summarize the experimental evidence. Don't give too specific, but then you can also refer to a specific figure table that was very illustrative and can really give support to that conclusion. And that way they can look back, oh yeah, I got this. Okay, if you jumped out to the discussion, you go back to that really good figure or table, and then they can understand. So if you're following Morrison, the book that we're really using here, he gave an example of how to write it out. It would be as this. The major conclusions of the study are that, and the conclusions are based on the following experimental evidence. First, we demonstrated that. Next, we provided strong evidence to support the conclusion that. Finally, then, and collectively, this experimental evidence strongly supports the overall concept that. So it's really easy, okay? I know. It's easy for me to say. When you sit down and write, it's another story, huh? Okay. But then this would be an example of opening, the discussion when you restate the major findings and how it led to the conclusions. So during the infection of gastric mucosa, H. pylori modulates the properties of host cells and tissues to generate a more suitable niche for colonization and persistence. In this study, we demonstrated H. pylori dependent modification of the abundant and important nuclear protein PARP1. Okay? So then they go to how contrary to original hypothesis and so forth. But other people like to give alternative would give a little bit more of the big picture perspective. So they would give a little paragraph of saying, giving the big picture first with your citation and so forth. Don't let it be too long if you want to do it that way. And then your next paragraph would be, our results indicate that the important plasma membrane SM, so forth, okay? So decide on how you want to start and then, stick to it. But don't let it be too long. Okay. So after the first paragraph, what do you do? There's a module style that you can think of the discussion as a collection of modular discourses or treatises, where every module will focus on one result, and then you use that to drive, the major point within that one paragraph, one module, or one topic per module. So that initially maybe you got 5 to 10 in the initial draft, and then you sort of like narrow it down and break it down and summarize it. So you get 3 or 4 modules in the final manuscript. That's one way to go. It would look something like this, which you start beginning. You just start paragraph 1, what I'm going to write, paragraph 2, paragraph 3, paragraph 4, paragraph 5, etc. And you get the main point. That's one way to go. And then where all these modules, you connect the dots between them, and with your current results, your current paper, and those of the previous papers, and then how these make it clear how these results relate to the current model. So does your work support or refute the accepted model? And then you point to the future. It's always important to say what you take from here and what you get up to future work. But then, but if you go back to Morrison style, which I thought was more specific, I like that one better. Your second and third paragraph would be to really give a succinct, really shortened review of literature and look at everything, all the evidence that's consistent with your conclusions. So you include your own prior publications and findings, if it's applicable, and how they supported the actual conclusions. You use published studies from other people and how they are consistent and complementary to yours. So this is like how we are all alike, and we all find the same thing. And then the next one would be the next paragraph, your fourth, actually. Oh, okay. So in this one, usually you can devote 1 or 2 paragraphs. So this is your second and third. Try not to go too much beyond that because it becomes too long, okay? So then on your fourth paragraph, then you can talk about the inconsistencies. What evidence is out there that's inconsistent with your actual conclusions? So you can give enough detail so you can really reasonably explain why it could be different. And some examples will be, well, in this study they use in these 2 studies they use different animal species or strains of mice, or the patient enrollment criteria was different. Or this study they used population in one country, and this other study is heterogeneous population in another country. Or this one study had post-surgical patients in an ICU, and these other patients are really with hematological malignancies. So really, this is like compare and contrast, really explain the contrast between the 2 different studies and maybe explain why they're inconsistent with yours. But when you're doing this, don't really start giving out negative comments about the other papers. You might think it was porqueria, right? But you don't say it. You don't write it, okay? Because you want to dish out the other authors, or you want to dish out other investigators. So in closing, what you want to do is make the case that why this conclusion would be justified, and despite the disparate findings reported by others. Okay, then next, what you can do is explain how these conclusions actually serve to advance the field, okay? You expand on what you already presented in your introduction, and then you go on to say, well, how do these conclusions move the field forward? What can be accomplished now that couldn't have been before? Well, maybe you found a new way to test a specific outcome, right? And then from there you say, okay, with this new way of doing this, you could probably do experiments on such and such populations and find if the outcomes come out differently, okay? What new questions have arisen? Maybe you found out that actually the gene that determines such and such disease wasn't the one you thought it was, and it was something else. They said, okay, look at that. Maybe the RB gene is the one that really is important when you're studying such and such disease. So we'll look at that more. So from here, what could go on from here? And how could they be approached always experimentally? That's really good. If you sort of like give a hint, and actually these papers are really good. If you're looking for ideas of what to do, find these papers that really give hints or really give out information on what needs to be done next, because maybe you can be the researchers that's doing what they're saying. Probably the group has already done it, but you never know. Maybe they haven't. Maybe the postdoc left the institution, they're not being done, so you could probably do it. So it's very good to do. And how filling the knowledge gap will help to advance the field, okay? And then for the 6th paragraph here now, we're addressing limitations. You have to be, science should always be transparent, right? Because all the ethical issues we learned from the HIV in the beginning and so forth, everybody from now on has to be very transparent. So what you want to do is you address what the limitations were regarding the conclusion and experimental design. So maybe they were already mentioned when you're trying to talk about the conclusions, how they differed. And then if you're really candid and realistic about this, you can really, really have a better chance of having the paper accepted, and you achieve credibility with your peers. If you're the first one to criticize yourself, then others won't do it, right? But if you sort of like try to just hocus pocus and not mention the limitations, people will find out and will express it. So you just express the limitations. You propose, other studies that might help others to become, how it could be figured out. And this will really, really help the desire of the reviewers to really ask you, which was the worst scenario, to repeat or do more experiments to be able to have that paper published, okay? You don't want them to tell them. So you just tell them, oh, look, these are my limitations. Maybe this can be done some other way or some other things. And, you leave it out there for other people to pick it up, okay? But don't let them make you say, okay, do a whole different set of experiments, because by that time you're saying, oh, I don't really want to do this, and forget the paper, okay? You don't want that. Okay, so then the final paragraph, as Manu has been saying, you always tell a story. When you tell a story, you need to have a satisfying ending, okay? So, in this ending, what you want is to repeat the major conclusions of the study and their significance in advancing the field. So this significance, that's why I put a caution sign to remind me, it must be crafted in such a way that resonates with the readership of the journal. If it's a specialized journal, really pick to them what they want to hear. If it's a cancer journal, talk like an oncologist. If it's a general biomedical journal, talk like a general scientist, okay? So you have to think who's going to be reading this and who cares about what I'm writing, okay? So the discussion should effectively tell a story about what was the scientific question that needed to be answered, what the actual answer to that question, the approach used to generate the answer to that question, and the reason why answering this question was important. If I tell you that, doesn't it sound easy? Yeah, all of you are in shock right now, huh? Okay, not easy yet? Okay, we hope it will become more easy if you answer these 4 items. Okay. So sometimes, and this is more so like in basic science, I guess, more than clinical science. Because clinical science, you really need to separate results and discussion. But in basic science, sometimes you need to combine results and discussion. So for your PhDs out there, I'm talking to you right now. When you find that each figure and table has to be explained extensively and compared to previous work, then maybe you need to combine results with a discussion within. Even some papers in basic science, you see them, they say results and discussion together, and then they have conclusions apart, where you get the take-home message there, okay? So if this happens to you, okay. But then also, if you think you don't have enough stuff to talk about in your discussion, then it might be a liability, so maybe you put it into the results and it doesn't look so empty, okay? So that's something to think about. Especially if you're doing something really novel that no one else has done before. Who are you going to compare to? If you're the first one to decide to do a certain experiment, you can't compare it to anybody. You're the first one. So what are you going to discuss about? Me, myself, and I? No. You have to discuss about other things. So maybe integrate it into the results, and then it'll look prettier, okay? So that's one thing to think about for you basic scientists. Then along those lines, a common mistake is, to begin with a detailed literature review that provides the rationale, and then you have 2 or 4 paragraphs that then you communicate the key findings. Remember I told you before, really have it up there really at the beginning so you can find it fast. Because then if you go into this long review of stuff of giving like the genesis of whatever, this, of this gene, who cares about that right now? Everybody knows the RB gene, right? So you don't want to go through all that. Because then at the final results, what you found out is it's not emphasized, okay? That you failed to recognize the importance. So don't repeat the introduction. And don't make it look like a review article. If you're going to write a review article, write it. But don't write it in your paper where you're trying to get new information out there, okay? So, so emphasis, remember that your emphasis is to communicate new knowledge, and it should be at the very beginning. Okay. Another pitfall would be if it's too long, as I explained. You're going off course, you're discussing issues that have nothing to do what you're talking about of your conclusion. There's no common thread. It goes off and off and off. You know, where's it all going? You have to be able to look back, step back, and look at it and say, okay, am I driving my point right here, or just going on and on and on? You don't want to be too dry either. Like, these results came out like this and this and this, and then it's boring, okay? You don't want to be bland also. I wrote these adjectives, by the way, so that I'm just creating here. You don't want to be bland because you want to you want to clearly communicate what the strength is and how it would be interesting to your readers. You don't want to overinterpret the results like, oh, look at this little point here, this is probably this, and this is this, and then no, that's too much. Okay, don't go beyond that. The data is the data. You put it there, it's out there, you show it to people, and that's it, okay? You don't want to speculate because it sounds like you're like this crazy scientist who likes to go off into space or something like that. But if you're really compelled because you're something that's very novel, you have no other people who to compare to, and you know, what are you going to talk about? You need to speculate, write it out. We speculate that this finding relates to a new undescribed phenomenon in, you know, gene expression. If you want to do that, okay, that's fine. But you wrote, you speculate. So then somebody else finds it out. Since you wrote it first, you get the Nobel Prize. How about that? Okay. Another pitfalls, and I wrote here, this is like going into the twilight zone. You don't want to inflate too much the importance of the findings. Please, you didn't invent science here, okay? It'll disgust the reviewers and the readers, and humility really goes a long way. You want to be over-eager. I like your results. Oh, I wrote this, me too, because you're like, oh, I got other results, but this big famous scientist got, so who cares? He doesn't care about you. What are you talking about? So the bully pulpit is those people who like to criticize and stand in a pulpit and criticize and attack other investigators or preach to the readers. No, this is not a church here. Okay, so then worst of all, drawing conclusions that are not supported by the data. You know, really avoid the temptation of allowing biases to go into your conclusions, okay? You have to stick to what was written there or was found there. Okay, now to give you some strategies. Get your readers to see the big picture, and where best to see it is Sistine Chapel, right? You have to look up and look at everything and see what's going on all over the place. How to win over the readers. A hook is, you can explain your results, how it probably potentially advance their work or provide new opportunities to your readers. They will like that. For people who are looking for ideas, that's the best way to look at those kind of articles to see what's best for you can continue studying. Okay, another strategy is like stirring the pot. Some people really like to do this, okay? So they want to find out there a disagreement or controversy in your paper, and oh, look, this finding differs from what this other famous person is finding. You know, want to stir the pot. But a reminder for those of you who like to stir the pot, you should know what you're cooking because before you can get burned, okay? So make sure if you're going to go that way, you might get burned or you might have to lick the spoon, as the saying says. So be careful. But some people, it's a good strategy because you're going to be sure that that paper is going to be cited a lot because you were the one who said that Mr. Fulano who's very famous, said, oh, no, you're wrong. I got this little study here that says differently. Okay? So but be sure you can handle all the backtake. Okay? Plugging the holes. It's very important to say, if you found a little holes, those little differences, and you said, yes, we thought of that, and we think we could probably do this and this to describe or further advance what's going on. Okay, advancing the field. Maybe if this is done a lot to people who change different ways, you know, to classify something. Different classifications, you know, if you propose new findings and then you as well, I know it's very distracting, isn't it, even to me? But anyway, we'll go on here. So if you really give out a new table and say, okay, we classified cancer for such and such a way, but with these new findings, adding perhaps the findings in genes and so forth, we're now going to classify this disease in this way. Those papers, no matter how boring they are, they always get published because you're proposing a new classification system. Okay, so if you find new findings, then maybe if you want to do that, that will be very good to publish, okay? And then, of course, be a visionary, and, you know, as Steve Jobs always explain what the future lies, because that will help you. They will know, people who read that will know that you are a thinker. You can envision what's coming up first, what comes up next, and then, you know, it will help other readers to understand it as well. Okay, so in writing the discussion, always write powerfully, be crisp and concise. And how do you write these beautiful discussions? Well, you have to write often. Be a practitioner. Write and write a lot until you get very, very good with your craft. And then I always say, whether or not you write well, write bravely. Just be brave and write. And then pester all your peers and everything. What do you look at this? Look at this. What do you think? Did it sound good? Did I get my point across? Does it? Do you understand it? And so forth. Okay, so that's where you get your feedback from many sources. Okay, so most of this stuff presented to you was from this book, which is very good if you wanted to refer for it, but we summarized it for you, so it's really good. And now is the part where if you have any questions. I think we turned out 15 minutes early, so we do have some time. Maybe you can answer a few questions. Maybe if you want to talk about what is the hardest part of you guys to really write, and, you know, have Matthew and I could probably give you a few tips. You know, what are the areas that you wanted to have more information on, so forth, or where do you get stuck? So maybe somebody could give feedback here. Oh, I think they're too much in shock to be able to answer. Okay, so, but, you know, I mean, what is the hardest part of you guys? I mean, how many say the introduction, when you have to get a good scientific literature review? That's most mentors say, start with the literature review and see how you go. Do you think that's really especially very hard? How many of you think? Yes? Or no? Or, yeah, I know it's very distracting. It's really driving me crazy here. But anyway. Hay que editar esto. Mariela, edita esto, por favor. Okay. But anyway, do you think maybe discussion is the worst part for you guys? Yes, okay. I found someone alive here, but thank you so much for that. You think the discussion is the worst, where you have to compare and contrast and everything. Now, do you think giving you specifically each paragraph what to address will be of help for you? Yes, okay. So, okay, I'm glad for that, because I think what you really need to do is, you know, a guideline to start, and then say, okay, maybe just, actually it starts with a sentence. You know, I have 5 paragraphs, and then say, okay, here I'm going to write limitations. Here I'm going to write, you know, comparisons. Here I'm going to write this. And then if you see the big picture, and you sort of like know where you go, and then you devote your time to like one little paragraph right here. Okay, today I'm going to write what are my limitations of the study. You get that over with. Remember, you have to draft it first, okay? So as you see, you know, pre-write, draft, revise, edit, and publish, okay? Then tomorrow I'm going to write about this stuff. And then when you get it all together, make sure it flows. You know, if you did it differently, make sure it flows one paragraph into another to make sure it tells the story as we've been telling you, okay? So that's really good to do, okay? And if I didn't used to do it before, but when I went through this book, it sounded very logical to me, because everybody tends to go, I'm going to write the introduction, I'm going to write materials and methods, and then I'm going to write the results, and then I'm going to write discussion. But I found very useful for Morrison to suggest differently, to get the results, because that's the exciting part. You want to really show your results, right? Like, oh, I got all this information. I want to do the graphs, and you want to do everything pretty in PowerPoint and everything and put it in there in your manuscript. That's the fun part. And then if you're fun and you're excited, go with the discussion, because then you're excited in that area. Then go back to your materials and methods and so forth. Other people really like to start with the introduction because that frames what they're going to. But it's all up to you. You decide your best way to do it, okay? Any other suggestions or questions or anything anyone wants to tell us? Or are we all okay here? Okay, so, well, thank you for listening to us, and I hope we've been helpful enough. And until next month, I guess, for part 3. Then we're going to be talking about the submission process and how to review. And these are stuff that not people know a lot about unless you've done it several times, okay? So it's really hopeful. It's not the good part, but it's the essential part to really have it published, okay? So good luck to all of you, and I hope you can.
There's a question.
Oh. Okay, so where in the binary should comment about detecting and removing handling outlier values and stats?
That you would do in your discussion.
Uh-huh.
In your discussion, you would talk about how you could also mention it in the methods, because it's part of your statistical analysis. How you handle outliers should be mentioned in methods, but you should also discuss it. You could even, it depends, if you have a graph where you actually show your outliers, well, you also mention them in the results. But I guess the answer is you need to describe how you detected these were outliers and how you decided that you were going to include or exclude from further analysis. So that goes in the methods. If you mention it in your results, it'll depend on if they're actually in the graph where you could actually show your outliers. For example, if you're looking at a volcano plot, that you're looking at expression, and you have these genes that are way out there compared to the rest of the genes, they're actually in your graph, and people are actually seeing them. You should mention them. And then maybe you could go in your discussion and address them as why they were there, or why you decided to use X and X methodology to identify your outliers to have them in or out of your study.
Okay, one last word. You're going to be sent out evaluations that you need to fill out, please. This is all of our process that everything needs to be evaluated. And once you fill it out, you'll get a handout of the slides given out today, okay? So be good with that. Thank you so much for your time.